Okay, so a little bit more about the molecular nature of what's going on here. Last time we introduced the Wnt beta-catenin pathway, and we talked about this signaling factor known as Wnt. It's a small diffusible factor, and it's going to land on receptors in the membrane and go through an intracellular transduction, that is communication of that information from the cell membrane to the cell nucleus, involving several proteins, including beta-catenin. And so beta-catenin initially is in a complex with several other proteins there in the cytoplasm. And either one of two things would happen. If no Wnt comes along, then this GSK3 protein here in the complex, that will stay stable, and it will take beta-catenin over and allow it to, let's see, another. So if GSK3 is stable, it takes beta-catenin over to this ubiquitin complex, which will degrade the beta-catenin. So beta-catenin will remain in the cytoplasm and eventually be degraded. On the other hand, if Wnt is present, then GSK3 will be inhibited with the opposite result. And that makes beta-catenin able to go to the nucleus. And here in the nucleus, it will interact with other factors and modify transcription of target genes. So that would actually complete the loop, if you will, from the extracellular through the cytoplasm into the nucleus, differential gene transcription as the result of Wnt signaling. Okay. So I think you can kind of logically remember this by, if the Wnt comes along, you're going to get a change in transcription down here. So for that to happen, beta-catenin has to remain stable and go to the nucleus. Yeah.
So by change in transcription, does that mean it like promotes transcription, or does it repress transcription? Oh, okay. So it depends on.
Yeah. I'm glad you brought that up, because that's it's something to keep in mind with differential gene transcription. Just means that it's different. It doesn't always mean that it's activating, because more than half the time it's repressing. And we tend to think of something about, it's more natural for us to think of proactive gene transcription as changing a situation. But in many cases, what the body actually does is to repress the transcription of something that had already started. Okay, so now we're going to see here a rapid reorganization, not just of maternal factors like dishevel, but of some of the cytoplasmic components such as microtubules. And in this figure, you can see microtubules that have been stained by a dye, and look at their organization. And these numbers here, 0.4, 0.5, and 0.7, they refer to the percent of the first cell cycle that has gone past. So from the moment of fertilization, and then 40% of the way through that first cell cycle, at that point the microtubules are basically all in a disarray. They're all overlapping with different directions. But then very rapidly, they're suddenly going to align in a parallel fashion that is parallel to the outside of the egg, or parallel within this cortex. And it's thought that factors such as dishevel are going to be carried along these microtubule arrays by microtubule motors. So in cell biology, you might have talked about things like kinesin and dynein, that are proteins that sit on microtubules or other structures, and they can carry cargoes such as RNAs or proteins from one place within the cell cytoplasm to somewhere else in that cytoplasm. And so dishevel, initially down here in the vaginal cap, is going to be carried along microtubules up towards the future dorsal side. And so here is a kinesin. It's a protein that is a microtubule motor, and it's bound to the microtubules. And then we have a couple of other guys, but dishevel essentially binds to that complex and gets moved along to a new location. In addition, there is also Wnt11 mRNAs that were present in the cortical cytoplasm. So just on the superficial layer of cytoplasm, these get moved to the new location too. So then up here in this future dorsal side, we're going to have Wnt11 mRNAs that get translated into protein. We're going to have disheveled and other things. And meanwhile, of course, the embryo is all going through cleavage, and these are becoming separated into daughter cells next to each other. And so essentially, we're going to be able to activate the Wnt pathway here on the dorsal side. On the other hand, the ventral side, there's not going to be Wnt11 mRNAs over here, so the Wnt pathway will not be active on the ventral side. I'm setting up a difference here in molecular terms between dorsal and ventral side. Okay. So you see how all these things tie together? The sperm enters, the cytoplasm moves, the factors move, cleavage occurs. So then, by the time we get to blastula stage, we're going to have beta-catenin will be in the nuclei due to this Wnt pathway activation. Beta-catenin will be in the nuclei on those cells on the dorsal side, but on the ventral side, beta-catenin will have been degraded because there was no Wnt pathway activated, and the default state is then to degrade beta-catenin. So if you look at GSK3, it's going to be inhibited on the dorsal side, allowing beta-catenin to go to the nucleus, but stable on the ventral side, causing beta-catenin to be degraded. So, once beta-catenin is in the nucleus, it's in the position, as we said, to engage in this differential gene transcription. And some of the target genes that it may activate are transcription factors. And transcription factors, as we know, these are proteins that will be in charge of regulating a number of different downstream target genes. So you can start with, say, mostly a single signal like beta-catenin, activate some transcription factors, and send out the minions to start getting some work done in this embryo. That is, start getting a lot of different genes activated or turned on or off, because that's what transcription factors do. And sometimes transcription factors will activate other transcription factors. So it expands, it amplifies. Now you get more and more regulatory factors contributing pretty directly to differential gene transcription. So this sort of series of events where you trigger an activation of transcription factors, and then that activates more transcription factors, and then more transcription factors. It's a pretty common paradigm for early developmental biology and the initiation of differentiation in cell fate. And you can sort of think of this like a waterfall or a cascade, as it's sometimes called, where a stream comes along and it initially falls over one little step and then kind of spreads out and falls over a successive series of steps as the river flows, and that's analogous to a successive series of steps for transcription factor families being activated and accomplishing a lot of changes in the gene products available to that cell. So, what is this mysterious nuclear center in molecular terms? And to find out what genes might be involved, maybe what are some of the targets of beta-catenin, you would look for genes that might be expressed in the right time in the right place. That's a totally logical place to start, right? Now if you're going to say, this is the critical transcription factor for taking the embryo to the next level, well, it probably ought to be expressed in the nuclear center. And it ought to be expressed in the young embryo at the time when the nuclear center is developing. So that might be one good way to, say, sort through a lot of different genes and get a clue about which ones are going to be the best candidates for taking the embryo to the next status in differentiation. Then once you have some candidate genes, you could try block it or move it approaches for those genes using any method that you want, and see whether or not, what kind of power those genes have to promote dorsal cell fate. And so if we think about beta-catenin in this context, if we block beta-catenin, we can see some result here. So first, let's look at the localization of beta-catenin. These two are immunohistochemistry experiments, so we're looking at beta-catenin protein and where you would find it in the embryo. The embryo is stained green just for background visualization. And at the 2-cell stage, beta-catenin is sort of more or less uniformly dispersed around the cortical cytoplasm. But then in the early gastrula stage, when the embryo is just about to begin gastrulation, now beta-catenin is more localized, more enriched over here on the future dorsal side. And then those two brown guys, those are also immunohistochemistry, looking at beta-catenin localization, and it gives you a little bit more of a nucleus versus cytoplasm view of things. And so the two embryos are just flipped around to show you dorsal side or ventral side when we're looking at it. And here on the dorsal side, these round spots, those are the nuclei, highly enriched for beta-catenin. Where then we flip the embryo around and show you the other side, there's no spots, so beta-catenin protein is more in the cytoplasm, or gone, definitely not in the nucleus. And here's a diagram of what we saw before, beta-catenin enriched in the nuclei in the dorsal side. So that's show it. Okay, block it. So in this particular experiment, to block beta-catenin, they took the approach of an antisense mRNA. And this is a reverse genetic approach, right? So we are starting with the target gene that we know about. We want to try to knock it down. And we're knocking it down at the mRNA level so that cells have transcribed beta-catenin mRNA. And we've then just injected into the cell an antisense for beta-catenin mRNA. So that the idea would be that the antisense, in a sense, transcripts would find each other, anneal, and then you could not get translation of that because the ribosome couldn't load onto that double-stranded RNA just for physical blockage reasons. And so it's a little bit of a crude method, but it does work to, say, eliminate the translation of your target RNA. So in this case, if you inject beta-catenin antisense into otherwise normal embryos that have been normally fertilized, they've undergone the cortical rotation, all that stuff, now look at this. You get a ventralized embryo. You get a belly piece. So just by blocking beta-catenin, it's as if we've obliterated the entire nucleus center. So beta-catenin must be a really critical factor within that nucleus center. You know, obviously not, well, probably not the only one. We didn't test that here. But we tested that this beta-catenin, is it necessary for dorsal fates to develop? Yes, it is. If you remove it, no dorsal fates. We could move it. Now we'll test if beta-catenin by itself would be sufficient to generate dorsal cell fates. And previously we know that in the classical days, Newcoop did this experiment where he arranged for the D3 blastomere to be have Newcoop components in it. So why don't we do this experiment where we inject the D3 blastomere with beta-catenin? So we'll still have our normal D1 blastomere over here, but we're going to introduce some beta-catenin on the opposite side, and that actually replicates the result of the duplicated head experiment, the result of the transplant at the D1 blastomere experiment. So that does support the hypothesis that this D1 blastomere, it supports the hypothesis that beta-catenin could arrange a nucleus center that would then be capable of promoting dorsal fates within the embryo. Any questions? Okay. So there was also this other classical observation. This experiment had been done and it could really never explain that lithium chloride treatment would dorsalize the embryo, would produce this two-headed phenotype here that we're characterizing as an overrepresentation of dorsal cell fates. And now that we know about beta-catenin, we could hypothesize what might be going on here. Do we somehow develop too much of the nucleus center signaling within the embryo? Would we, in fact, produce too much beta-catenin, now that we think beta-catenin is a very critical factor, both necessary and sufficient to establish the nuclear center. And in 2003, this was actually studied within the embryo. So some embryos were treated with lithium chloride, and then they went in and looked at the beta-catenin in the Wnt pathway to see what sort of effect that the lithium chloride was having upon the embryo. And sure enough, that's exactly what you would expect. So here's our control situation where beta-catenin is being degraded on the ventral side or enriched in the nuclei on the dorsal side. And when you treat with lithium chloride, effectively it inhibited the GSK3 everywhere in the embryo. And as a result then, beta-catenin becomes stable everywhere in the embryo. And then you have beta-catenin in the nuclei everywhere in the embryo, including on the ventral side. And that then went ahead and developed this two-headed embryo. Okay, question?
But the ventral side still developed anyway, even though it was everywhere. So meaning that stuff has no effect on creating the ventral side, just making another.
So are you saying this embryo has a ventral side to it?
Yeah, that just seems, because if you take it all away, you only get belly. But if you add it everywhere, you still get ventral too, you just get two dorsal.
Right, okay. Well, I would say this embryo has an overrepresentation of dorsal sulfates, and it does have some ventral sulfates in there as well. And you might speculate that the cells that have the highest concentration of beta-catenin might become the dorsal, and then those that have none or a little bit less would become the ventral. It's true, this is sort of everywhere, so
Just interesting.
yeah, I don't know how to explain that down to that level of detail, but in general, the idea that beta-catenin associated with dorsal sulfates is holding up based on this hypothesis. Yeah.
So it's more like you're getting an extra dorsal side instead of replacing the ventral side with another dorsal side in this case?
Yeah, I don't know which is more accurate to say,
Okay.
the extra dorsal side or replacing the ventral side. I think probably I would lean towards an extra dorsal side because you've got two sites of information, two nuclear centers saying build the dorsal side here. And that's like the dorsal side is the more proactive thing that you have to create, and the ventral side is the default.
Okay.
But there's always seems to be a portion of the cells that would go into the default. So you do get some. I mean, otherwise the entire thing would just become localized tissue. We're not quite to that extreme.
Okay.
Okay, let me see. I think we're. So shall we have a breakdown? We're 8:52, and let's go ahead and come back at. Let's just come back at 9 o'clock.